And here are my conflicts of interest. Now, ladies and gentlemen, the Canadian government has warned me they will not take any responsibility for the next slide I'm going to show. And I won't take any questions on this next slide either. Normally, the residents and the junior faculty tell about themselves, and I have not done so over 30 plus years, but you're going to hear it now. I was born in Canada. I was one of 4 boys. This was my family in Toronto. But I changed my name at age 13 to James Bond. I became a player. I played basketball day and night. We were the Toronto champions. I also liked to play football, but I loved to water ski. And I used to water ski every summer and became an instructor and taught at a camp for many years. Larry, hold your breath. I like to play guitar a lot. The Beatles songs. Met a woman, signed some papers, and 3 dogs later started to form a family. They're my daughters. One of them signed some papers. This was the result. And pregnancy came again, and on Monday, this came along. So I'm pretty happy right now. I was chief of orthopedics here for a while. I became a tenured professor with an endowed chair from the university. I spend a lot of time in the OR, and I figured out it's between 10 and 15,000 cases I've done. I spent a lot of time in clinic. This is an old slide, but you can see that it's a team approach, and that'll be one of the themes I'll talk about today. These are my lab members, some of them new, some of them old. We like to do a lot of research in the lab, and of course in the clinic, we like to have fun. And recently I was fortunate enough to receive the Lifetime Achievement Award from the Association of Bone and Joint Surgery. And like a good Canadian, the $10,000 check went into the department's funds to send these people to the Orthopedic Research Society. And I would encourage you, as all of you win awards, because you do, to donate to your compatriots to help send them to meetings. In terms of academic accomplishments, there's the standard blurb that I use with visiting professor, but here are some of the details. And I'd like, as you read this, to look at probably my most proud achievement. And that is I've won, like many of you here, the Saul Halpern Orthopedic Educator Award here at Stanford. You all are the most important thing in our academic lives, and you are our legacy, and I'm sure all the faculty will agree with that. So I'm going to talk about the scientific basis of orthopedics and what I've done, basically mostly in the lab. I asked my fellow if he knew any of the work I did in the lab and why people know my name, and he had no idea, which is, I guess, okay. That's why they spend a year here. But orthopedics is a combination of biology, biomechanics, and biomaterials. But it's also about mentors and role models. And I've been very, very lucky. Dr. Salter, you see, was the supreme clinician scientist. Next to him, Dr. Schatzker and Dr. Tile, who you all know for their work. Alan Gross, one of the premier orthopedic reconstructionists. Doctors Babechko and others here, Ian McNabb, spine surgeons who had a great influence on me. Dr. Hastings taught me a lot about juvenile arthritis. And The fellow at the lower right is Victor Bernazier. I had the good fortune to win a rotation with him for 6 months. He was an orthopedic pathologist. And I looked under a microscope and I dissected spines from people that passed away and did a lot of basic anatomy and pathology with him. Internationally, I've had a lot of great mentors too. Lars Lidgren, who many of you might know from the Bone and Joint Decade. It was his idea, and he told me about it walking along the Baltic at his summer home. And next thing I know, the Pope, the President, and the UN had all declared the Bone and Joint Decade. Per Aspenberg, my PhD supervisor, who was a tremendous artist. This is a self-portrait. And I did my PhD in Lund, Sweden. My opponent, as I've told Rosa, was the chairman of the University of Helsinki in Finland, and one of the smartest orthopedic surgeons I have ever met. He was also an Olympian. He was a speed skater. He kept in shape in the summer bicycling. These are the Rydholm brothers from Lund, Sweden. One was and still is the editor-in-chief of Acta Orthopedica. He went through my PhD thesis with a red pen. I've never seen so much red in my life. That's Anders. And Urban had the largest Swedish juvenile arthritis clinic in the country. This is James Anderson, who Lane Smith knows very well. He's probably the most famous person in the world. He's a pathologist. He knows more about inflammation and the foreign body response. And he is in Cleveland at Case Western. Very good friend, Lane and mine. And all these people are supermen and superwomen. There are women who I haven't shown pictures of, but there are many of them. But what they've emphasized to me is that orthopedics is a team approach, whether it be research, teaching, or education, or research. So Dr. Anderson taught me about inflammation. This is his classic article. And there's a biological response whenever you injure someone, whether it be by surgery, trauma, or otherwise, where cells in the vicinity, macrophages and others, sense what's going on and send signals on. And there's a progression of cells that come in and signal other cells, and hopefully this all resolves. So we've come a long way from juvenile arthritis and the implants which we use today, because this is before my time, but at least on the top, you see the Judet acrylic prosthesis, a resurfacing, hemi-resurfacing, the ring prosthesis, McKee-Ferrard, those two are metal on metal. And I remember having to take out the one on the top right as a resin. And I'm telling you, this thing was so far up into the pelvis. It was osseointegrated. It was hard to get out. And I've shown you implants, all the mistakes that surgeons and manufacturers and researchers made over the years, not so much with polyethylene, but you can see all the suboptimal implant designs with points at the end and hard edges that crack bone cement, etc. Now, the person who changed all of our lives was Sir John Charnley. And he did much of this work in his garage with minimal resources. But he noticed, as I show with that yellow pointer, that in some cases, there were large areas of bone destruction. And working with Dr. Bernazier in Toronto, the pathologist, Joe Schatzker had done a lot of resurfacing arthroplasties that Heinz Wagner from Germany had shown him how to do. And this is with the old polyethylene, and it's a big head. It was a wear machine. And these, I think he did about 250 of them. By the time I was his resident, a few years later, we were taking some of them out. And they had this tan-colored foreign body response that if you look under polarized light on the bottom right, you see these yellow, positively birefringent polyethylene fibers, and you can see the foreign body response, all the giant cells. Well, this was chronic inflammation and foreign body response. So chronic inflammation is the infiltration of these mononuclear cells, mainly macrophages, some of the others, ongoing tissue destruction, but also repair. And it looks like this. And I'm glad I'm following a pathology lecture because you're all tuned up. You can see there's a lot of cells there, a lot of inflammatory cells. The bigger cells are macrophages, and you can see some small blood vessels here. So there's inflammation going on with repair. That's chronic inflammation. Now, particle disease as we know it today actually started out as cement disease because we would cement all the implants. And in fact, cement disease was described by Lynn Jones and David Hungerford. I was just on the phone with Lynn probably an hour ago. And you can see the massive areas of osteolysis around the hip replacements, most of which were cemented. But you can see the polyethylene wear and the arrows around the large holes around the knee implants, which I've pointed out here. So osteolysis isn't all about hips. It's about any joint that has polyethylene. And we see this at surgery. This is a good example of polyethylene wear. This is a revision I did some years ago. Now, all of this was described by Professor Willert in Germany. Willert was a pathologist and an orthopedic surgeon. And what he found was that if you generate particles, usually the body can take care of things. And you get sort of a homeostasis between the generation of wear particles and the body's ability to deal with it. And you don't get osteolysis. But if something goes awry and the inflammatory reaction continues, you get holes in the bone because these are inflammatory cells that release cytokines and chemokines. They tell cells to come in, and next thing you know, you have huge amounts of osteolysis. And I can tell you, many of us for a long time did a lot of revisions for wear. We don't do as much anymore, but I'll show you a few that have come in recently. I was very fortunate enough to meet Professor Willert. He had a poster at a meeting and he died shortly after that. But we had some long, great discussions about osteolysis. So I started doing a bunch of research here at Stanford, looking at this membrane, growing it in culture with Lane Smith and Dr. Maloney, and a lot of fellows from all over the world who came to work in Lane's lab. And we showed that this membrane wasn't this sleepy type membrane. It was very dynamic. It was making all these pro-inflammatory factors that were degrading the bone. Well, about 10 or so years later, I went to Sweden and I had a sabbatical and I combined my research with Per Aspenberg's research. That's the guy who did the self image in Lund, Sweden. And I began research work there and at Stanford. I had rabbits and rats there and here, would fly back and forth because I still had a full load here. But I did a short time there and there were word processors, but no real computers. So I used to sit by the fax machine every night to get my papers from Per Aspenberg that he would correct and send back and forth and back and forth. And a lot of it, this was done with this chamber. This is called the bone harvest chamber. This chamber actually was invented in Gothenburg, Sweden.
Okay. If you put in titanium alloy particles, okay, this is 10 particles per mil, not much happened. So I went up to the 9 particles per mil, and and we got nice suppression. Now, this is in the 1990s now. Look what happens if you put in cobalt chrome particles. No bone. Necrosis, terrible cheap.
Had a lot of debris, and here's one you can see, very subtle, but you can see the osteolysis in the proximal femur here. It's radiolucent, and here it is, and he had tremendous pain, a big effusion, and needed a big revision to get the thing out. Now we are still seeing cement disease and polyethylene disease too. This is a case I did probably in the last 6 months. So people are living longer, so it's still relevant. And it takes a lot of resources, a lot of help, and I'm grateful to the residents and the fellows and the nurses for helping me through these difficult cases, because they're long, they're arduous, and sometimes things break and the outcome often isn't what you want. So particle disease was waning. We still used it as a method to study inflammation. We coated our particles with LPS, which is the membrane of E. coli, and we studied inflammation. But I remember what Tommy Lasorda said about the only problem with success, and we had some good success, is that it doesn't teach you how to do with failure. Now, we didn't have failure, but we had sort of waning interest in particle disease. You have to retool. And that's one of the themes. And Mike Gardner and I were just talking about this, about retooling. And I remembered my years of doing trauma, not only as a resident, I helped set up the Level 1 Trauma Center here, and grateful to Dr. Lowenberg and the others in the trauma section here, that bone loss and dealing with bone problems is not just about osteolysis. It's about how to deal with these difficult fractures, non-unions. On the right, you see infection that's left a huge critical size defect and a gap. And these are not easy to deal with. And when you think about bone healing, which is at the bottom, the bone cells, you think of the MSC, or the mesenchymal stem cell, or stromal cell. But if you look at bone healing, the first stage is inflammation. I said, oh, I know a lot about inflammation. I've been doing this for 20 years now. So, and then I realized as I was doing my revisions, I couldn't keep putting more metal and plastic back, because that's not the right thing. As Alan Gross taught me, put money in the bank. Put bone back. Try and reconstruct bone. Because what you're doing now is going to fail, and you have to be prepared. So I started studying bone healing and the interaction of bone healing cells, MSCs, osteoblasts, osteoclasts, and inflammatory cells. And we know these inflammatory cells from work done by many people, including at Boston University and others. And if you look at the cytokines that occur after a fracture happens, you see these are all our good friends, inflammatory cytokines, TNF-alpha, IL-1, etc. And it's not till later that you see BMPs and TGF-beta and everything else. So that sort of validated our concept of inflammation and bone healing. And if you skip acute inflammation, the bones don't heal. I wrote a big grant on this, trying to skip inflammation. It was never funded, thank goodness, because it would have been a failure. But you need to go through inflammation. Why? Because the cytokines and all the other inflammatory factors, they activate or license the stromal cells, the MSCs, to do their thing. And in fact, this is part of the innate immune system. Whether you have a bacterium or you have bits of cells that have broken down our nucleus. They all are noticed by receptors on macrophages and leukocytes of different types. And they do all of these things. They release pro-inflammatory cytokines, chemokines to tell cells to come in, growth factors, they activate T cells, so you get that, and you also have resolution. And I thought this was original, but it wasn't. If you look at the Nobel laureate George Bernard Shaw, in The Doctor's Dilemma, now this is 1925, he said, there is at the bottom only one genuinely scientific treatment for all diseases, and that is stimulate the phagocytes, stimulate the phagocytes. Drugs are a delusion. Way ahead of his time. And when I went to medical school, macrophages ate things. They were garbage eaters. But in fact, we know they're part of the innate immune system, probably at least as important as the T lymphocytes. And over the years, it's not just been pro and anti-inflammatory macrophages, M1s and M2s, but now it's a huge spectrum. And Professor Mantovani in Italy was really responsible, amongst others, to help us with that. And we did studies to show that if you grew macrophages of different types with pre-osteoblasts, whether it be taking the supernatants that they're grown in or actually co-culture them, you got more bone. Macrophages and osteoblasts like each other. And in fact, if you change the M1s, the pro-inflammatory, to the anti-inflammatory, pro-reconstructives, you have to wait 3 days. You got more bone in culture, more brown at the bottom if you wait a few days. Inflammation is important. We've come to realize that all of these cells are constantly talking to one another. There's modulation of all of these cells on each other. And it depends on timing. For acute inflammation, if you have too much of it, of course, the animal dies, including humans. If you don't have enough of it, then you don't activate the MSCs. But in chronic inflammation, the persistence is not good, and you want to quell that. And that's what we've been doing for the last number of years. We've tried to modulate macrophages coming into a site, changing if they're pro or anti-inflammatory, modulating their transcription factor, NF-kappa B, and also we've been putting genes into stem cells to try and do things more than they would normally do. And we've been genetically modifying them. And I'll just go through this quickly. With Fan Yang, coated a rod with anti-microphage chemokine to keep them from coming in, and we published on that. We made an MSC, a stem cell, a mononuclear stromal cell, and we put in a gene for an anti-inflammatory cytokine. But more importantly, we hooked that gene up to a sensing gene so that when inflammation was high, NF-kappa B was up, it would spew out IL-4. But when it was down, it would stop. So it was much like the thermometers and the temperature controls that you have in your room. And we published on that. And now we're into a new area. And I'm very grateful to my collaborators, Brice Godelier and Chuck Chan and many others, for taking me into the single cell realm, because that's where science is right now. We have this mouse model of a critical size bone defect, and it works. And this was in collaboration with Stefan Zwingenberger from Germany. And we can modulate how these fractures heal by putting in an anti-inflammatory cytokine, which you see here, IL-4, the thing heals. But you have to wait 3 days, because if you put it in right away, you're going to inhibit inflammation. We don't want that. We're looking at, on a single cell level, what cells are there over time. And we're doing something called cytoph, which is mass cytometry, where we can put these cells through a cell sorter, and we can find out with this very sophisticated method which cells are near each other and which cells are talking to one another. And interestingly, if we put in MSCs into this defect, we can track them. The majority of these MSCs don't produce the bone. They talk to local cells that produce the actual bone. We have single cell RNA sequencing, looking at the RNA and what it's doing. And we're looking at, again, what's expressed, what cells are there, and looking at specific pathways. So cells, we can dissect out which cells are talking to which and which pathways are the most important ones. We still use our pump model where we put in particle contamination into that pump, and we study inflammation and how we can modulate inflammation. And in fact, we have found that if we can put in an NF-kappa B, the transcription factor for inflammation, an inhibitor of that, we get more bone forming. We're doing a lot of work on osteonecrosis with Peter Yang. Peter and I and his fellows have designed a physiologically, mechanically relevant, bioabsorbable, functionally graded scaffold so we can decompress, put in this scaffold to guide cells into the osteonecrotic area in patients who have a round femoral head, not a collapsed one. They need total hip replacement. And we've been putting in these genetically modified MSCs to help all of this along. And in conjunction with the University of Pittsburgh and the University of North Texas, we have an NIH grant also on this joint on a chip. Now, I don't know if you know, but Europe has decided there's going to be no animal research in a couple of years. Now, I don't know if it's 2 or 3 years, but it's coming up. So they'll cell culture, fine, other methods too. So we have this grant. There's at the national, at NIH, there's different groups. And there's like kidney on a chip and heart on a chip and lung on a chip and everything on. So we have the grant on joint on a chip. And what we do is we have bone and cartilage, we have synovium, and we have adipose tissue, and we have macrophages, of course. And we connect them all up into this system you can see on the top right, and we can continuously provide flow into this area and harvest these tissues or the supernatants ever so often and simulate different types of arthritis. And the idea here, this is with human tissues. And so you can do a high throughput, high screening method of looking at different drugs. And of course, it has to be validated, which we've done with animal models and human tissues. And now we're in the drug screening. So we can screen a lot of drugs quickly. And we've done work on validating the system with IL-1 beta, which is one of the chief inflammatory factors with arthritis. And you can see that we have markers of which are well known in osteoarthritis to validate the system. And now with a new collaborator, we have added neurons because synovium and the joint has neurons, and we added vascular cells too. And now we have the new mini joint, which has neurons, and we're doing that now. This is what it looks like. We can keep this going for about 6 weeks. Why are we doing all of this? Well, I think that orthopedics, the way I was trained, and I was trained in Toronto by Salter and all these clinician scientists, I think the way to understand what we're doing is to start with the basics. Start with biomechanics, the stuff that Dr. Giori is doing. To start with biology. And Lane Smith has ushered a new era here of biology over decades. And biomaterials, which Fan, Yang, myself, and Peter Yang, and others in bioengineering and in mechanical engineering and material science are doing. And that way, we can get closer to understanding how things work before we implant them in humans or even animals. And I'd like to end with this quote, which I like very much. It's again by another Nobel Prize laureate in physics. And Werner Heisenberg, heard of the Heisenberg principle, he really put quantum mechanics in our lives. And he said, you have to remember that what we observe is not nature in itself, but nature exposed to our method of questioning. Now, I've been very fortunate. Our lab has had funding for many, many years, and hopefully, if the gods are good to us, or if you're an atheist, someone's good to you, then we'll continue. And I might add, it was only this morning I got an email from NIH. We had put in a prospective, multi-center, randomized study with about 10 or 12 centers looking at osteonecrosis, core decompression, with or without cells, and all the biology part. The mass cytometry is done here, but it's all over the whole country. It was just approved for funding. So it's a very big move, and I think it'll probably answer a question that many of us have asked. But I want to thank all the many people who have helped our causes over the years, the people in the lab and those who've been in the lab, and the list of pictures which I've had here, which actually are only partial, those at Stanford, those in other departments at Stanford, across the world. We have collaborators in Hong Kong, in Finland, in Sweden, in France, in Germany, Czech Republic, Japan, China, etc., etc. So with that, I thank you very much for listening. Be happy to take some questions.